clinical applications and the legality and all of that business before we move on. So as we mentioned, anandamide is the actual neurotransmitter. The system wasn't really found until 1992. Functions, and the reason it has become clinically important in many states, although not Utah, although there actually is a pill form that you can take, which is called Marinol, but we'll talk about that in just a second. So it's involved in the CNS and the PNS and pain signaling. So I want you to understand pain signaling. So as far as pain management, the management things that we have talked about are pain sensation, right? You can stop it here where you're sensing it. You could potentially stop it in the signaling on its way to the brain, right? Either in the PNS signaling or CNS, right? Signaling. And then you can do the awareness of pain in the brain, right? And opioids work on the brain, right? And these work instead on the pain signaling, getting it to the brain. So can you see how this would, at least theoretically, be a pain management technique? Yeah? In addition, I want to reinforce that, go the other, that this does not tap into the dopamine pathway. So the whole drug-seeking behavior and the real chemical addiction that's reinforced by dopamine, the endocannabinoids do not do this, right? Which means that they don't have that particular risk. Now, are there any negatives to this drug being used for pain management? There are negatives to every single drug you've ever found, right? There is no perfect drug. We know the negatives to opioids. We know that, of course, opioids can lead to death because of depression of the respiratory system. We know that they lead to chemical addiction. And now let's talk about the negatives associated with using endocannabinoids or using THC, which are basically THC in marijuana is an anandamide agonist. So let me show you what anandamide looks like, not that you'll care. It's a lipid-soluble neurotransmitter. It's the only one we've talked about. It's not the only one that exists, but it's the only one we've talked about. Now, that's not particularly important to you, but now I want you to see the similarity in the 3D structure of anandamide and THC. Can you see why THC is an anandamide agonist, why it actually fits into the receptors for anandamide? It's a pretty weak anandamide agonist, but it's lipid soluble, so whatever you take in gets there very quickly. And even if it's weak and it falls off, right, you've got more to bind, right? Does that make sense? So it's a very weak anandamide agonist. The other reason that it's really, really effective, it has an effect almost instantaneously, is because even though we didn't know anything about the anandamide system or the endocannabinoid system until 92, we have figured out that you have receptors for anandamide everywhere, all over your cerebrum and your cerebellum. You've got them in your peripheral nervous system. So if you take in a lipid-soluble neurotransmitter, and it actually, there's no limitation to where it can go because it's lipid-soluble, right? If you take in a lipid-soluble drug that mimics the neurotransmitter, it can go everywhere. And even if it binds loosely, it's going to have massive effects, right? So it is pretty effective very, very quickly. What are the negatives associated with using THC for pain management? There are negatives, right? Otherwise, well, all states would have it, probably except Utah, because we wouldn't, right? It'll happen, but Utah's always 50 years behind the rest of the country. So what are the negatives? Can it kill you? No, nobody dies from marijuana, right? Is it chemically habit-forming? It's not chemically it is habit-forming. I mean, you can find people who just smoke weed all damn day and get fat and boring, but it's not chemically habit-forming, right? So, so it doesn't tap into the dopamine pathway. So what are the negatives? Yeah, weight gain it does increase your appetite, so weight gain is a possibility. Sometimes that's what you want when you're dealing with a chronic disease, though, right? With dealing with cancer or fibromyalgia, then what else?
Maybe lack of concentration.
Yeah, it actually does impact your working memory, not your long-term memory formation, but when people are using THC, so let's say it's medical, so that people don't get pissed off about me using this example. Somebody got so mad that I actually talked about anything positive related to drugs last semester. She's like, I cannot even believe that you talked about it. I was like, you just wanted me to lie to you, right? But you have friends for that. So she was like, I can't believe you said anything positive about alcohol or whatever. It's like, I'm a physiology professor. I'm just going to tell you what like is in the literature. So with working memory, what marijuana tends to do, or THC in marijuana tends to do, is like you can sit around and you're like going to have brilliant ideas, and then like honestly, within the next 15 minutes, you will have no idea that you had them, right? It's just like you completely forgot. It's like you when you're really distracted and you walk into the kitchen and you're like can't remember why you went there. That's like every moment when you're on marijuana. But if you've got fibromyalgia, maybe that's the best damn day you ever had. Honestly, it's like I'm gonna forget for a minute that everything hurts all the damn time. So the working memory issue is an issue. Eating and sleeping patterns and pleasure and food rewarding. So it's not without its negatives. Why did it become illegal in this country anyway? Does anybody have any idea of the history of marijuana? It has to do with a really strong core of racism. They were super, super afraid that Hispanic people and African American people were going to use this to grab on to our Vestal virgin white women. And yeah, so go look at the history of why a lot of drugs are illegal because of like a really strong history of racism. But now we can dump that and we can reassess it with the science. And it is still, however, federally, you know, that there's differences between the federal and the state laws. It's legal in a lot of states. It's still federally illegal. They're not prosecuting it very often. But if you cross state lines, even if you have a medical marijuana card, it's still illegal to cross state lines with what you have, right? Now, because we've been able to study it, we need to know, now, since 1992, we had discovered that there's at least 2 categories of receptors for it. They're called the cannabinoid receptors, CB1 and CB2. One of them has more to do with pain relief, and the other one has more to do with pleasure. And so now, of course, now that we can research it in other countries, it's completely legal, like in Canada, you can actually grow strains that are more specific to either pain relief or anxiety relief or this or that. You can even grow strains that don't make you feel the pleasure, right? So you get pain relief but very, very little pleasure. And I'm not quite sure why we really want to make people who've been in pain for a long time not have a pleasant day, but that's what people are comfortable with. So you can grow those strains as well. So the receptors are CB1 and CB2. Now, they are among the most abundant CNS receptors and PNS receptors we've ever found. So they're all over the brain. So what does THC do? THC is a weak anandamide agonist. It binds, it falls off, it binds, it falls off, it binds, it falls off. And since it's lipid soluble, right, both THC and anandamide are lipid soluble, you have multiple ways that you can take it into the body, yes? So you can ingest it, right? You can inhale it. You can use patches for it. You can pretty much do anything that allows it to get into the body via lipid soluble mechanisms. So there's a bunch of different ways of taking it into the body. In Utah, of course, both medical and recreational is still illegal. However, there's a pill that you can get called Marinol. And Marinol is basically the derivatives without all the pleasure. And the problem with Marinol is that when it's taken in this pill form, you have to up the dose so much before you actually get the effect. And it also means that instead of getting the effect within 2 minutes, it's about 30 minutes to get the effect. That hasn't been particularly useful at pain management. In states, I just went to the CDC and found this stat yesterday. In states that have legalized medical marijuana, in 2 states that have legalized, that have been keeping stats, not everybody has, opioid overdoses have gone down 20% since they legalized medical marijuana. So it's at least a conversation that we should have. You can disagree with me if you want, but we're all going to be voting soon, right? And you don't have to agree with me. I want you to know where to find reputable sources. I told you guys about scholar.google.com, right? Okay. So if you want to find a reputable source, just a second, I'll get you. Go to scholar.google.com, and then if you want to really critically evaluate any kind of information, what you need to do is, of course, not find it in one study, right? Anything can happen once, but if you find it repeated in 10 studies, and they're peer-reviewed journals that we're talking about, then it has more credibility. And then look at sample sizes, right? Are we talking about 2 people, or are we talking about 20,000 people, right? And then you make educated decisions, even if it means that you vehemently disagree with your mother-in-law, right?
So how would it, so if you were to give them like medically THC or whatever, how would that go against their opioid addiction if it
I'm not saying it's a treatment for opioid addiction. What I'm saying is it is a treatment for pain. And so a lot of people who get into opioid addiction pathways got into it because of pain treatment, right? So this doesn't do anything to the dopamine system. Now, if that person could get off of opioids, this might be a replacement analgesic, right? It doesn't do anything to that system. Though. Go ahead, Rejoice.
So we have anandamide receptors everywhere, everywhere in the body. Is there a possibility that they can bind to like the wrong receptor, like?
Oh yeah, totally. Remember that this hasn't even we haven't been able to study it. So do we know everything about anandamide? Absolutely not. Do we know everything about long-term usage of THC? We don't. So, you know, and that's a lot of times, in Utah, for instance, that's the common argument against, is that the research hasn't been done. But we're sort of in a little conundrum there, because as long as you don't legalize it, then the research won't get done, right? And when the research isn't done, then it can't be used, right, and legalized. So we've got a little feedback loop there that we're going to get past it. Utah will probably be among the last states to get past it, with the exception of the fact that, did you guys know, with the last time they had this referendum on legalizing the oil derivatives of, I can't remember what it's called. Yeah, cannabis oil. What?
CBD.
CBD. They were. It almost passed. In part because there was a huge lobby of moms, huge, powerful lobby of moms, mostly Mormon moms, honestly, that this works for neurological disorders when most things don't work. And so, for instance, it is a treatment for childhood epilepsy when damn near nothing works, except, for instance, using massive quantities of benzos, right? So massive quantities of benzos with kids with neurological disorders will work, but that has some serious side effects. And this is more effective. So it almost made it. The oil almost made it. But of course, that's an interesting one too, because it's not legal to produce the oil here in the state, and it's also not legal to transport the oil across state lines. So you just have to magically get it somehow. So I don't know. It'll be interesting, but I think it's worth you guys knowing, because you're going to vote, right? And you're going to like figure out how the hell you want to vote, and you may contradict my vote. You know how I'm voting. I don't even like this stuff, but I think people who are in pain should have some options, man, other than opioids. And I also don't think it's my business if you guys are going to, like, you've been in pain for all your life and then you're going to have a good day. I think that's great. You have a good day. You have cancer. You have a good day. Okay. So the other thing that we figured out about anandamide, and anandamide is interesting because anandamide actually, I don't know who the hell thought of this study. I would like to know the history on it. But anandamide has been figured out, and also THC, to inhibit the growth of breast cancer in vitro. So we're talking about in the laboratory when you're growing up breast cancer cells. Both anandamide and THC will inhibit the growth of breast cancer. And so now they're using it in animal models to see if we can move it into human models. We don't know why it works. I wonder how somebody came up with that study in the first place. Maybe it was just like an accident. Whoops, spilled my weed. I have no idea. I don't know how they did it. Like, I like to know the process of science. Like, what is the story behind it? I don't know the story behind that. And then they figured out that, of course, all of us are junkies. Half the reason you like cocoa and chocolate is because there are weak anandamide agonists, think very similar to THC, in cocoa and chocolate. That's like, oh, that was super pleasant. That's because you just got a little high. That's why. Okay. But that's an acceptable one here in Utah. I wish that if I could go back, and nobody is going to let me, and if I could go back and assess the legality versus illegality and availability of drugs, it had something to do with physiology instead of with history and anachronisms and racisms and, fake news, all of that kind of stuff. Just like, let's look at the ones that kill you. I promise you that I would not make meth legal, promise, right? Heroin, still be illegal, right? Cocaine, still illegal, right? But there are a few of them that we could reconsider and figure out. Okay, so the last two are these miscellaneous neurotransmitters. I don't care that you know what these do. I think it's just kind of interesting that they are like neurotransmitters that we figured out lately. So carbon monoxide, if you breathe it in, it will kill you dead as a hammer. And it happens every single winter in Utah, and even more in Texas. Because Texas, I don't know why, every winter people are really, really surprised that it gets cold. And it gets super cold in Texas. It's like super extreme. It's really, really hot. And they're like, I'm super surprised and I didn't have my heater checked. And so what do? Because everybody's got a barbecue grill. They bring the damn barbecue grill inside the house. We should probably let them secede, honestly. And then people die of carbon monoxide poisoning. Now, so carbon monoxide is a killer if you breathe it in, but your brain apparently makes it and releases it as a neurotransmitter. WTH. I don't get it. And then nitric oxide gas, not nitrous. That's the giggle gas at the dentist. We all like that. So nitric oxide gas is bubbled up and released as a neurotransmitter. And there's a lot of study into this one because this one's the erectile dysfunction neurotransmitter. And you know how much money people will throw at making sure they still get boners? All of it, like all of my money, just have it all, right? So that one has a lot of research. So that one's nitric oxide. It's a PNS neurotransmitter. It also works in the CNS in some places. Okay, so last little tidbit is just to talk about receptor functions and why receptors are named the way they are and all of that kind of stuff. So it's not consistent, right? Because a cholinergic receptor is named for the neurotransmitter, but then an opioid receptor is named for the drug, right? So there's no consistency with how the receptors are named. So how do you know? Well, let me just tell you the story, and that is that you don't know. It has to do with what was found first. So often the impact of a drug is known long before we even knew there was an endogenous chemical or a receptor for that endogenous chemical, right? So we talked about opioids being used since 3400 BC and cannabis being used since, what was it, 2700 BC? 2700 BC, long time. So people used those long before they knew about the neurotransmitter system. But conceptually, here's something that makes sense. The receptor and the endogenous chemical had to predate the drug, meaning that you don't have opium receptors in your brain. Yes? What you have is receptors that respond to an endogenous chemical and respond to opium. Yes? Does that make sense? So the receptor system had to predate whatever chemical we're using. Does that make sense? So you don't have an opium receptor. So there are receptors that bind to an endogenous chemical and to opium, which made me come up with this funny thought that I totally love, and I'm not going to test you on, but that means that if we think, okay, my background's in evolutionary biology and molecular and evolutionary biology. That means that, so you know that, for instance, a poison dart frog evolved the capacity to make a chemical which is basically keeping things from eating it, right? And you probably know that, for instance, some seed dispersal mechanisms are, I have this delicious fruit around the seed and then something's going to eat it and then I'm going to walk around and poop out the seed and then that's how the seed dispersal mechanism works. Well, my little hypothesis that I came up with that I find fascinating, and I'm just going to torture you with, is that opium is using us as a dispersal mechanism. Cannabis is using us as a dispersal mechanism. Cannabis is using us to make sure that there is the survival of cannabis. And opium is using us, I mean, not consciously, right? Opium is using us to make sure that there will always be more opium plants. And it's incredibly effective, isn't it? Like, are we going to run out of opium poppies anytime soon? No, because what do? We're like, we took that, we're like, that was delicious. Harvest the seeds, grow some more. That was delicious. Harvest the seeds, grow some more. And so the population of opium poppies has exploded all over the world. The population of cannabis has exploded all over the world as a little convergent evolution in which, we are actually, manipulating, right? So I think it's kind of interesting. Basically, what we're doing is doing the opposite of basically keeping something from eating you, right? We're making sure that you ingest it, and then basically with agricultural revolution, we're just going to keep making it. So if the drug was found first, then a lot of times the receptors were named for the drug, like opioid receptors. But if the chemical was found first, then a lot of times the receptors are named for the endogenous chemical, like cholinergic receptors, GABAergic receptors, dopaminergic receptors. Unfortunately, there is no one system. It's just all historically, however anything was found. Okay, so that means that when you are a medical professional and you find an opioid receptor, you will remember, okay, this one binds to opium, but there's also a chemical that it binds to, right? Okay, so now we're going to do a little bit more about receptors. Nobody likes this part. You probably won't like it either. It's fine. You have to do it anyway. I don't even really like teaching it, honestly. But okay, so now what we are going to talk about, and I need you to get the big picture before we get lost in the details. So what we have talked about many, many times is, okay, I want to say I'm a chemical messenger and she is my target cell, yes? And you guys are all her homies inside the cell, right? So she's the receptor, yes? And this is the rest of the cell membrane, and you guys are all of the organelles and intracellular molecules, yeah? So what's going to happen is we already know that the chemical messenger can bind to the receptor and then cause an effect in the cell, yeah? But what we never talked about is how. How do you cause an effect in the cell? What happens inside the cell? It's not poof, it ain't magic. Because if you look at anything hard enough, it ain't magic, right? So what we have to figure out is how you cause an effect. And what we do is we call these signal transduction pathways. I'm a signal, and she has to transduce it. Did you guys ever play telephone as a kid? Okay, this is like playing telephone. She is a cell membrane receptor, yes? So she has to stay there. But she needs to get something to happen all the way to the dude in the back. Okay, he's the nucleus. I need for the nucleus to do, for instance, transcription, and then make me an mRNA so that I can have a new protein, right? And then I can have a new function. So what I'm going to do is I'm going to bind to her, and she has to stay there, and she has to make something happen to you, and then you have to make something happen to you, all the way to get to the back, right? So this is called a signal transduction pathway, yeah? Now, let's just talk about that telephone game. With that telephone game, which type of chemical messenger is going to have to play a longer game of telephone? A lipid-soluble chemical messenger or a water-soluble chemical messenger? Yes, because water has to stay outside. If I am a lipid-soluble chemical messenger, I can go straight inside the cell and to the source. Yes? Does that make sense? So the signal transduction pathway is basically what happens in between when you bind to the receptor and the cell has its response. Okay? So these are signal transduction pathways, and they dramatically differ depending on whether you're talking about lipid-soluble or chemical or water-soluble. So let's start with the easy ones first. Shortest game of telephone will be intracellular receptor. I'm going to find my receptor intracellular. But intracellular receptors are only for blank soluble chemical messengers. Lipid soluble chemical messengers. So if I use an intracellular receptor, I don't have to play very much telephone at all. I can go all the way through, and I can actually cause directly what I want to occur in the cell. So what I am going to do in there, and I've got an animation for you. This figure's from your textbook, but we'll also look at an animation. So I have a lipid-soluble chemical messenger, and it goes straight through by diffusion, and it can diffuse through the plasma membrane because it's lipid-soluble, exactly. So it can diffuse straight through, and it can either bind to a receptor on the cytoplasm or a receptor that's in the nucleus, okay? When you bind to the receptor, it doesn't have to be a hormone. That's just the type of chemical messenger that they use. This chemical receptor complex has a different shape than either the chemical or the receptor alone, yeah? And then that new shape actually can go and bind directly to the gene of interest on the DNA and start transcription of that gene, yeah? Get an mRNA, and then do translation of that gene, and then I'll have a new protein. And a new protein might be a new structural protein, it might be a new enzyme, it might be a new metabolic protein. Does that make sense? And that's how you can change the activity of the cell. Okay, this is called gene activation. So let me show you an animation for gene activation. Okay. This one also uses a hormone. The only reason these are using hormones more than neurotransmitters is because there's boatloads of lipid-soluble hormones. And we haven't found very many lipid-soluble neurotransmitters. And we think anandamide works this way, but we're not quite sure yet. We haven't figured out the intracellular mechanism for anandamide yet. Okay. So this is aldosterone, which is a lipid-soluble hormone, and it's going to go straight through the plasma membrane and find its receptor, the intracellular receptor. This one just happens to be in the cytoplasm. Go straight through the nuclear pore. You remember there's pores, right? Okay. And then it's going to go and bind to a specific region of the DNA to activate transcription, which is production of an mRNA from a DNA transcript. And then the mRNA is going to go where for translation? Rough ER or a free ribosome. Either way, a ribosome, right? That make sense? So we are going to, if it was meant for secretion, it would have to go to the rough ER. If it was meant to stay intracellular, it could go to a free ribosome. Okay, now this new protein, I don't know what it does, something different, otherwise we wouldn't have bothered, right? Okay, could be structural, functional protein, metabolic protein, whatever. Does that make sense? So that is called gene activation, and gene activation uses intracellular receptors and lipid-soluble chemical messengers. So examples of chemical messengers that use these. All of the steroid hormones, including all of your sex hormones, do this: estrogen, progesterone, testosterone, and then some other hormones. And we think that there are some neurotransmitters that use this, but we don't know much about the lipid-soluble neurotransmitters yet. Okay? All right. Now, this ends up being important, right? Because if, for instance, I needed to take an estrogen antagonist or an estrogen agonist, did you know that if someone is, for instance, taking in testosterone, like, okay, so let's say you're a bodybuilder and you're shooting yourself up with testosterone, don't do that. But testosterone actually gets converted into estrogen, FYI. So if you want to have pecs and not boobs, what do you need to take? You need to take testosterone and an estrogen. Yeah, and it's actually an estrogen antagonist. It's an estrogen blocker, which means that you had to know that this was an intracellular receptor and figure out how to bind to it, right? So there's a lot of pharmacology that goes into receptors, tons of pharmacology that goes into receptors. And by the way, lots of people who take testosterone to build their muscle mass are not the sharpest knife in the drawer. And they do not realize that they should probably also be taking an estrogen blocker, and then they're going to get pecs and boobs. Go ahead.
What are some estrogen blockers? Because they make like complexes for that stuff. How do people take?
There are actually prescription estrogen blockers that you would give in a medical situation, and they are just called estrogen antagonists. I don't know any name brands of them. Does anybody? Go ahead.
Oh, no.
No? So I don't know any name brands of them. But if you are getting the black market stuff, you can take both, right? And they actually do have complexes of both of them, but they're prescription only or black market only. It's not over-the-counter stuff that you would be able to get. Go ahead.
For lipid-soluble stuff, I need to be co-transported with something else to not make it lipid-soluble?
No, we want it to be lipid-soluble. So you just give it in its lipid-soluble form, and it goes just everywhere it wants to, but it doesn't have any impact if it doesn't find a receptor. Right? So a lot of times they'll actually have to provide more of it. So you have to have like more testosterone than you would actually need for it to get to its receptor. Okay. And we're going to talk about those whole like testosterone and all of that business and bulking up with testosterone supplements and why it makes your testes shrink to the size of English peas. And make you super pissed off, and then your girlfriend dumps you, and you're like, oh, I wanted muscle so I could have a girlfriend. You're like, she said you were an asshole. So so we'll talk all about that when we get to endocrine system. Okay, so that was the only way that you guys are going to learn. It's not the only way that exists, but it's the only way that's really common for lipid-soluble chemical messengers to bind to the receptor and cause the effect. Okay, there are other ways, but they're not super common. Okay, so now what we're going to do is water-soluble chemical messengers are way more common. And so you are going to learn 4 different signal transduction pathways for those. And the signal transduction pathways are basically going to be longer because I have to stay outside the cell, right? Okay, so they're a little bit more complicated. You can totally do it though, and you actually already know one of them. You just don't know that it fits into this category. So I'm going to deal with a plasma or cell membrane receptor, right? And it's going to stay stuck there. I'm going to be a water-soluble chemical messenger, and I'm going to have to stay outside the cell, okay? So the types of cell membrane receptors and signal transduction pathways, the first one just starts with a ligand-gated ion channel, but you've totally seen those before, right? So how do I get this cell to do something? The chemical messenger binds to and opens, or you could close, a ligand-gated ion channel and therefore lets ions in or out that wouldn't otherwise have been let in and out, right? And we know that because that's the really common way for neurotransmitters to work, right? You can open a sodium channel and depolarize. You can open a potassium channel and hyperpolarize, right? So what we are saying is the signal transduction pathway inside the cell is started because you bind to an ion channel. And the signal transduction pathway is basically an action potential in that one, right? Okay, so how do you get something to occur? You bind to an ion channel and you change the electrical nature of the membrane. FYI though, that's not the only thing that you can do to a cell by binding to an ion channel. You can, for instance, bind to an ion channel, let something in, let's say calcium. Calcium is about 300 times higher in the extracellular fluid than in the intracellular fluid, so it will move in really readily. What if letting calcium inside the cell made the cell behave differently by, for instance, activating an enzyme that wasn't active before? The calcium binds to it and activates it, right? Or inducing exocytosis. You've seen an instance in which intracellular calcium concentration increasing causes exocytosis, right? Or what if it activated a pathway that ultimately led to a change in metabolism or protein synthesis, right? Does that make sense? So either way, you start the whole process by opening an ion channel that wasn't open before. And that is how you start the signal transduction pathway. That good? Okay, so this is a water-soluble chemical messenger, and the receptor is an ion channel receptor. So let's see some examples of that you might care about. Nicotinic cholinergic receptors are ion channel receptors, specifically acetylcholine binding to a nicotinic cholinergic receptor will open a sodium channel and cause depolarization, right? And you find those in the central nervous system, and they're important in learning, attention, and memory. Does that make sense? You will also find nicotinic cholinergic receptors in the entire next chapter, because we are going to be dealing with neuromuscular junctions with skeletal muscle. They all use acetylcholine. They are all nicotinic cholinergic receptors, and they all are sodium channels, right? So we are going to run into a whole chapter of examples of those, okay? So skeletal muscle cells are nicotinic cholinergic receptors, or have nicotinic cholinergic receptors. Okay, you guys all right with that? I mean, you already knew that one. So that's the first way that a water-soluble chemical messenger could activate a cell activity. The second one is, what if instead of the receptor being an ion channel, this time the receptor is an enzyme? So look up here for a second. Okay, so chemical messenger binds to receptor. This side that faces the extracellular fluid has a receptor binding site and functions as a receptor, but the side that faces the intracellular fluid is an enzyme. It's all the same protein, but it's got a receptor side and an enzymatic side. So what happens when you bind to it is you activate the enzyme. The enzyme was inactive before. So when I activate the enzyme, I can change cell function. I can change metabolism. I can make proteins happen, whatever the hell I do, right? Ultimately, this starts a little cascade, right? A little telephone that will get me a new cell activity by activating an enzyme, okay? This specific type in which the receptor is the receptor is part of the same protein that is an enzyme. This is a big category of receptors called the tyrosine kinase receptors. And the reason they're called the tyrosine kinase receptors is because what they do is they phosphorylate, kinase, the tyrosine amino acid of a protein, and that's how you start the activity. I don't really care that you remember that specifically, but they are called tyrosine kinase receptors, and what they do is the receptor side and the enzyme side are part of the same protein. You bind to it, and that's how you activate cell activity. Okay, so why do you care? Because a lot of hormone receptors for water-soluble hormones are tyrosine kinase receptors. Do you think that if you ended up going into something related to OBGYN or endocrinology, you would end up dealing with tyrosine kinase receptors? You totally would, right? Right? Anything related to endocrinology would have to deal with these receptors. You can't find the drug if you don't know what the receptor looks like. Yeah? Okay, so tyrosine kinase receptors, one more time. One side of the same protein is a receptor and the other side is an enzyme. Okay, now the next set looks very similar, and for some reason your textbook did not put a picture of it in there, which is so weird to me because they're like among the most common types of receptors. So let me show you. This is very similar to a tyrosine kinase receptor, just one difference, okay? So this is a tyrosine kinase receptor. I've got a receptor side and an enzyme side, but they are part of the same protein. With me? Okay, these are the tyrosine kinase receptors. They activate this enzyme, and that's how you get cell stuff started. The only difference with this next category is these are called JAK kinase receptors. The receptor and the enzyme are two separate proteins, strings of amino acids, but they're stuck to each other. Okay? Now, they're going to have a different shape, so you couldn't use the same drug with the two of them. But what does it do? It does the same thing, right? Something binds to the receptor, and that's how you activate the enzyme and change the cell's activity. Is that okay? So these are called JAK kinase receptors. Why do you have to know these? Because they are so damn common that, get this, JAK kinase means just another kinase. They found so damn many of them, they got tired of naming and numbering them. When I was in graduate school, we did molecular biology, and with the protein lab, I did the DNA lab, but with the protein lab, they would go, oh, I found a kinase, and they're like, oh, great, did you find hay in a barn too? Because there's like so many of these. They're like super common receptors. And literally, they used to name them after themselves and think they were going to get a Nobel Prize. And now you're lucky if you like get a blurb of a publication for finding a new JAK kinase. So these are really, really common receptor types. And they end up being really important in a couple of places. So again, the only difference is instead of being one string of amino acids, this is two different strings. Binding to that will activate that. So growth hormone binds to a JAK kinase receptor. Now, of course, we have to figure out how to make growth hormone in the lab, and it has to bind to the receptor. So we had to know what the receptor looked like, right? So now if, for instance, some child is born and has pituitary dwarfism, and we want to treat them with growth hormone, we give them an artificial growth hormone that has to bind to the right receptor. So a lot of clinical treatments are based on receptor types. Okay, last one, hardest one, weirdest one for me to understand are called the Gprotein coupled receptors. And so let me just tell you about this. And, okay, so the Gprotein coupled receptors. What this is the most indirect way of causing a cell response that I could possibly imagine. Because they basically play telephone on the cell surface before you even start the telephone inside the cell. So with this, what happens is, if, for instance, I'm thinking, let's think we're playing pool for a second, okay? And the ion channel is the pocket. The direct way to do it would be to shoot directly in the pocket, like using a membrane ion channel, right? And you just shoot straight at it. You basically just have the cell membrane, the chemical messenger bind directly to it, right? But instead of doing that, I'm going to use a combo shot that ends up using 1, 2, 3, 4 different balls before I actually hit the ion channel. Okay, so I've got an animation for you and I'll describe it to you. I literally cannot imagine how this was an evolutionarily stable strategy at all. It seems like there's so much that could go wrong. So what I'm really curious about is how far back in evolution do you find these things? Because look at how they work. Here's what happens. This is your textbook's picture, which I kind of hate for one reason, but I got an animation. Chemical messenger binds to a cell membrane receptor. This is the thing that I actually want to open, but I ain't anywhere near it. So what happens is chemical messenger binds to the receptor, changes the shape of the receptor such that these 3 other proteins slide through the phospholipid bilayer, and one of the subunits smacks into that one, and then that's how you activate it. What? In Texas, anybody else, I know I've got another one Texan here. Mary, you're a Texan, right? Oh, 2? In Texas, maybe it was just my mom. My mom, when I did something by the least efficient way possible, she'd say, Mel, why the hell are you going around your ass to get to your elbow? That's what this is doing. You're going around your ass to get to your elbow. Why 72 steps when you could, did you guys know that expression? You do? Oh, you're the first one I've ever heard. Okay, good. So in Texas, that's how we talk, and we think it's charming. And in Utah, apparently, I think it is. Okay. So you're going around your ass to get to your elbow. But the reason this is confusing is because they actually put the alpha subunit over here and then show it over there. That's dumb. Why are they called Gprotein coupled receptors? Because the energy for this is from GTP, guanosine triphosphate, instead of adenosine triphosphate. So now let me show you an animation and it'll make it make more sense. I mean, kind of make more sense. Okay, so what we have is, okay. This guy, right? Stop doing that. Okay, hold on. What we have is this guy. Okay, so here's the G proteins. Alpha, beta, is that gamma? Alpha, beta, is that gamma? Okay, alpha, beta, and gamma. What's going to happen is they didn't put the protein that you want to activate in this picture, so I want you to imagine that it has to smash into this guy to actually activate it, okay? So what happens is the water-soluble chemical messenger, hormone or whatever it is, or a neurotransmitter, or whatever, is going to bind to the cell membrane receptor, and then that changes the shape, right? And then you have to use GTP to allow for the subunits to come off, and then at some point, the alpha subunit dissociates and slides by itself into that other protein, and that's how you activate it. This protein doesn't have to be an ion channel. Hold on just a second. This protein doesn't have to be an ion channel. It could be an enzyme, and that's how you activate the enzyme. Does anybody else have the question of how the hell that evolved? Like, this looks like there's so much that could go wrong. I would think that there would be no life on this damn planet, right? It's like, why would you have 5 proteins when you could do it with 1? But it did, whatever. So I want to know how far back it goes in evolution. So then what happens is you will use GTP again to get it to go back into position so that you can use it again. So I'll hydrolyze a phosphate off of GTP, guanosine triphosphate, get it moving, and then all of it will go back. That make sense? I have no idea. It's confusing as hell to me. When I first learned about that, I was like, what? I didn't even believe him. I'm like a biology professor was telling me this how it works, and I was like, I don't really believe you. That sounds dumb, but it does work that way, and it's so damn important that like this is it's used. These are the Gprotein receptors are the muscarinic cholinergic receptors for acetylcholine that are used for learning and attention and memory in the brain. So that's important, right? Okay. They are the alpha adrenergic receptors that you use for norepinephrine and epinephrine to change blood flow patterns to your skin and your viscera. And then this is how you might have heard of them even without knowing you were hearing of them before. The beta adrenergic receptors. Have you ever heard of beta blockers? Yeah. Okay, so a beta blocker, the beta adrenergic receptors on your heart, when norepinephrine or epinephrine binds to them, it increases cardiac muscle contraction. But if I have heart disease or high blood pressure, what I want to do is slow down the heart. So I use a drug that is a beta adrenergic antagonist and blocks the effect, right? Which means I had to know what these work like and what these look like. Right? So these are beta blockers. The beta adrenergic receptors on the heart are these G protein coupled receptors. That's how they work. So it's pretty freaking important, right? Okay, so that's how beta blockers work. And then serotonin, like half of us would like more serotonin in our life, right? Those are G protein coupled receptors as well, right? So it's pretty damn important, even though I can't understand what the hell's going on there, right? I just eventually trusted him. I was like, and then I just wrote it down on the test and got an A, and I was fine. So I asked him that question. I was like, can you tell me why this, give me a story that helps me understand how this evolved? And he was like, we are moving on, Melanie. We are moving on. I was like, whatever. Okay. Okay. So, now we're done. But before we go we're done with that. We've got one last thing to do on this part. So before we go on, I want you guys to, without looking at your notes, start hearing the words that will be in questions about this. And also know that every question I ask about this, people miss it left, right, and center. And I think it's just because it's wordy and it's kind of hard to understand and you don't like studying it. So what is the signal transduction mechanism for lipid-soluble chemical messengers that you learned? What's it called? Gene activation. Yes. Okay, that makes sense? Okay. Now, there are 4 signal transduction mechanisms or pathways for water-soluble chemical messengers. Yes? The first one that you learned was simply starting the signal transduction pathway with an ion channel receptor. Yeah? And you know examples of that, and we've seen examples of that. The second one was starting the signal transduction with a receptor that is also an enzyme. What are those called? Tyrosine kinase receptors. The third one is starting one with a receptor that is bound to an enzyme, and those are JAK kinase receptors. And the fourth one is going around your ass to get to your elbow to get something started. And what's that? Gprotein receptors.
Okay, have to word it on the test just like that.
I'm not going to, but if you write it and you also come up with Gprotein receptors, I'll give you credit, and I'll also smile. I'll be like, look, I'm a missionary to a foreign land. I'm bringing my words of wisdom. That's what I always figure. I was like, you guys need more of me here. You got plenty of you. Okay. So what happens at the end of all of this signal transduction? Well, basically, I told you, there's a physiological change in the cell, otherwise you wouldn't have bothered. This is not a comprehensive list, but what could you do? You could activate transcription, cause the synthesis of proteins, either directly with gene activation or indirectly with one of these telephone games. You could activate an enzyme, deactivate an enzyme. You could induce secretion or cell division or depolarize or hyperpolarize or change the metabolic rate. But I promise you, you did something. Otherwise, you wouldn't have bothered. Okay? So those signal transduction mechanisms, by the way, are broadly for everything. They are for paracrine and autocrine. They are for neurotransmitters and hormones. They are for all of those things. They actually are way back in chapter 5, but can you see why I didn't teach those to you during the first 2 weeks of class? Because I didn't want you to drop. And now it's after the drop date, so neener. Okay. So now, last little bit is just something interesting to sort of bring all of this back around. So what I want to talk about is that the nervous system actually has the capacity to subconsciously, and also consciously in some ways, decide what is worth more action potentials and therefore more ATP, and what is worth fewer action potentials and therefore less ATP. So you guys all know this, and we've talked about it before. So whenever you're trying to get an action potential, we know the beginning. We know that you can't get an action potential normally in the body, unless we shock you. We can't get an action potential without starting with a graded potential, yes? And graded potentials can be triggered by damn near anything but electricity, right? Light, temperature, pressure, pain, chemicals, whatever. They can be triggered by any of those things. And then we know that if you get a sufficiently depolarizing graded potential to hit threshold, then you get an action potential, yes? And we know that in a cell, one action potential is just like any other action potentials. There aren't strong or weak, right? And we also know that the way that you code for intensity in the nervous system is not by the magnitude of an individual action potential, but it's by the what? Frequency of action potentials over time. So now we need to talk about this concept of how does your body, brain, and subconscious actually decide what is worth frequent action potentials and therefore expense, right? And what is worth infrequent action potentials. And the concept is called adaptation or accommodation. And so what we actually do is we actually tend to attend, right? Like either consciously or subconsciously to certain categories of things and also to new stimuli. So what happens is you have the capacity to sort of decide how many action potentials to spend on a given stimulus. So let's talk about sensory stimuli because that's the easiest thing to talk about. Okay, so right now you are attending to my voice, right? You are deciding to attend to my voice, and you are not attending as much to other things, but it's super low level. Like for instance, right now, you can feel your clothes on your body. Now attend to it. You can consciously decide to feel your clothes on your body, right? So what you do when you attend to it is you tend to increase the number of action potentials that you actually devote to it. And most of the time, all of that happens subconsciously. So why are you, for instance, paying attention to the sound of my voice versus the clicking of a pen or the humming of that thing or the freaking construction that's always going on around us? Why do you do that? Well, what your body does is generally what happens is any constant stimulus, if it is a relatively constant stimulus, it doesn't seem to be worth really frequent action potentials, especially since the first time I encountered it, what I generally do with the first time I encounter a new stimulus, I either consciously or subconsciously categorize it, right? The feeling of my clothes on my body when I first put them on and I was naked, I could feel them, right? You can feel the clothes on your body. And sometimes it's like wool and you're like, ew, you have to take it off. I can't wear wool at all. Gross, it makes me smell. Does anybody else smell weird when they wear wool? I literally smell like a sheep. It makes me sweat so bad. I don't know how people wear wool. I'm like, that's so pretty. I'll never wear that. So you put it on, and then what happens is you're taking in these stimuli, and they're constant. And if they're constant and they don't fit into the category of something that could kill you, something that you could eat, or something that you could have sex with, then basically you're just like, it's fine. We don't need frequent action potentials on this. Okay? So what I want you to think about is I want you to think about our ancestors walking across the African savannah, right? And they are walking across the African savannah and they have all kinds of sensory input. what you have is, of course, light, but you're also, for instance, hearing the water go through the stream and hearing the wind go through the rushes, and you're hearing the birds tweet, and you're seeing all kinds of stuff. And so you're not paying a ton of attention to those, but if you attended to them, you would know that you had been hearing them the whole time, right? Okay. And then you hear, even if it's not loud, right? What happens? You attend, and you attend to new stimuli. Does it make evolutionary sense to you that new stimuli are worth your ATP? Yes. So you attend to the new stimuli, right? And then you have to categorize it, either consciously or subconsciously, and it happens almost instantaneously. Can I have sex with it? Is it going to eat me? Can I eat it? Right? And if it fits into any of those categories, quite often you attend to it for quite a long time, right? Okay? If it's like something super juicy and I want to eat that, you're like, oh, I'm going to attend to that. Or if it's something's going to eat me, or if it's like the most sexy thing I've ever seen in my life, and I'm going to attend to it, right? So with that, what happens is you attend to new stimuli, but it doesn't have to be loud or super disturbing. How many of you guys are parents, right? In Utah, quite a few of you, oh my God, this is the least parenting class I've ever seen. In Utah, honestly. Like in Texas, nobody was parents before 27, except for the couple of people, right? But in Utah, it's like, I've got 12 children. So I am 25, I have 12 children. But at least you were a child, right? If you don't have a child, you were a child. So I want you to think about sitting in a room, or your mom sitting in a room, and you're sitting in a room and you're doing your own stuff. You're doing your parents' stuff, you got crap to do, you're farting around on the internet, you're watching a television show, you are making something, whatever, you are doing whatever. And your kids are in the other room, and you hear them, and they're making kid noises. You know, things are falling over, they're like squawking at each other, there's a video game going on, somebody's annoying somebody else. It's all just like kid noises, right? But then you hear silence. What happens? Oh my God! It's a change in stimulus, and it's as powerful a change in stimulus as if you heard the bookcase fall over, right? So we are set up to notice changes and to send very frequent action potentials with new stimuli. And then if they are constant, what happens is we become relatively insensitive at our sensory receptors, and then we send fewer action potentials over the same time period. But we can choose, because we have a cerebral cortex, to attend to things in some ways. Like I can choose at any moment right now to attend to the feeling of my clothes, or to attend to the sound that's outside. I can do any of that with my cerebral cortex. So we're set up to basically pay attention to new stimuli, and we spend a lot more money, ATP, on new stimuli. And then last but not least, so what happens is that when stimuli are constant, you cease to consciously notice them at all. So for instance, have you ever driven through an entire town that smells atrocious? Like a whole town. Like if you were from the South, you have driven through paper mill towns. They smell like butt foot. The entire town smells like a sock was used to wipe somebody's bum, and then they like are waving it around. It's terrible. And I don't know how people live there, but now I do know how people live there, because what happens is that stimulus is constant, right? And you no longer attend to that stimulus. Does that mean that they can't smell a fart? No, they totally can smell a fart or perfume, because that's going to be a new stimulus. Does that make sense? So the same thing happens with you guys with constant stimulus. And then the last thing is that males and females tend to be different in their capacities for adaptation and accommodation. And this is interesting to talk about. Now, I'm not a complete sexist. I am a feminist, but I'm not a complete sexist. I'm not a sexist at all. So these are like bell curves, and I'm not telling you that any man or any woman could not fall anywhere on the bell curve. But based on evolutionary biology, before about the last 150 years, if we're talking back to hunter-gatherer societies and even agrarian societies, males and females had pretty different roles, historically speaking, right? So let's think back to hunter-gatherer societies and realize that we've been dramatically different from those things not very long in the vast scheme of things. So if we think about males and what the typical male pattern would be, and we tend to be relatively social groups in humans, but males tended to do things like this. They were going to, for instance, be the one who would go out and hunt the wildebeest, right? kill the wildebeest, drag the wildebeest back home, feed himself first, feed his family second, have sex, go to sleep, do the same thing the next day, right? So it's a series of single focus activities. And on the bell curve, males tend to be better at single focus activities. They adapt or accommodate out extraneous information. So when I go up to my husband and he's playing a video game, that's his wildebeest, man. And I am the wind through the rushes. And I say, hey, babe, so-and-so invited us out to dinner tonight. Do you want to go? And he's like, sure, yeah, let's go. What time? 7? Sure. And I ask him later at 6:30, he's like, you literally never told me that. It's because I am not the wildebeest, right? Right? So there had to be sex or fear, right? Or food. And those things work way better. I have experimented. But women tend to be on the other end of the bell curve, in which that, historically speaking, we were more likely to be in the social group, right? And we had more kids than we could keep alive, and more social interact. Honestly, really, they just died. They just like fell off of something, some ate them, whatever. So got a disease. So we had a lot of things to attend to in our social interactions to attend to, in addition to children, in addition to caring for everything. So women tend to, on the bell curve, be better at multiple focus activities simultaneously, which means that they're worse at attending out extraneous information, right? So if, for instance, my husband comes and says, hey, baby, let's go. And my mother-in-law is coming over in 6 minutes, right? And the children are screaming and the sink is dripping. There's literally no way in hell that he's going to get my single focus activity, right? Because I'm hearing everything. It's also the reason we go out to dinner together, and I can talking to him, and I can hear all 3 conversations that are going on around me. It's like, how can you hear that? It's like, she is pissed. It's super funny. Males and females tend to be a little different on this in their capacities for adaptation and accommodation. Okay, so you got to be the wildebeest, ladies. You got to be the wildebeest. Go ahead.
So what about people with neurological disorders like autism? What is happening? They're not really able to put out other sensory.
Yeah. So just so you know, autism So I can give you some interesting resources. My family has a huge, deep history of autism. My husband's family, actually. What we know is that people on the spectrum tend to not be very good at adapting or accommodating out extraneous information. And so excess sensory information tends to be really disturbing to people with autism. What we don't know is why. We don't know what's going on. We do know that there are at least 6 portions of the brain that are different in people on the spectrum than they are in people who are neurotypical. What we don't know yet is which portion of the brain has to do with that whole adaptation accommodation. But I've got some good references for you if you want to read into it. There isn't an answer yet. There are a lot of really good questions and some good suggestions from the preliminary research, but we don't know. So my husband's family, I don't know if you guys know this, but you're statistically more likely to end up on the spectrum when your family are mathematicians and engineers. And my husband is from a very long history of mathematicians and engineers, and autism is rampant in his family. So we have, his brother is autistic, his uncle is autistic. Every single firstborn child in my children's generation is either on the spectrum or just off the spectrum, like every single firstborn male. So there are 4 in this generation that are on the spectrum or off the spectrum. Really strong genetic component, really strong behavioral component. And it's not like it being an engineer makes you an autistic, but the predisposition to that sort of thing ends up manifesting in autism, especially if you get 2 engineers together making a baby, right? Yeah. Then you get, yeah. So yeah, I've read a ton about that, and we still don't know all the answers, but there's some really good questions out there. Okay, so next thing we are going to do is going to correlate to what you guys are doing in lab this week, and that is we are going to do muscle physiology. And you guys are doing muscle physiology in lab this week, and it's actually a fun lab. I like that one. We're working on improving the physiology lab, and hopefully this one will actually be good for you. If you get to be the person that gets shocked, volunteer. What? What were you going to say?
No, we just did that yesterday.
Oh, you did? Good. Okay. So what we will do on Tuesday is we will put together with a lot of the stuff that you did in lab. We won't have enough time to get to it actually today, but we'll get started on it today. So let me tell you first off, you're going to have a quiz on Tuesday, and it's just going to be over synapses, not muscles, right? So a quiz over synapses, and we're going to start muscles today. But you can't do the P in muscles unless you remember the A. So you have to go back, and I'm not saying I need you to go back and remind yourself of the name of all the damn forearm muscles, which is what I call them because I hate them, the damn forearm muscles. But you do need to know the concepts of, for instance, antagonistic muscle pairs, right? How do you get a joint to move two different ways, right? And then you need to know the basic gross anatomy of a muscle all the way from the big to the small, right? A whole muscle is wrapped in epimysium, and then you get fascicles, and then you need to get all the way down to the point that you can talk about the structures of an individual muscle fiber. And remember that it's annoying for all of us, but they renamed every blasted thing inside that muscle cell, right? You can't even call it a plasma membrane anymore. You got to call it a stupid sarcolemma. And then not cytoplasm, but sarcoplasm. So all the way down to the actin and the myosin. If this is problematic for you, you need to go review it because you will have a hard time with the physiology if you don't remember the anatomy. Okay, so today we won't get super far into it. You've got time to review it before that. Okay, so your objective will be to, before I see you on Tuesday, be able to build a whole skeletal muscle from the gross to the microscopic, right? Know those. All right, so if you need help with the anatomy review, interactive physiology is really good for that too. Okay, so let's just start talking about the functions of skeletal muscle generally, and then we'll talk a little bit about the structures. We won't get too far today. So when we start talking about the functions of skeletal muscle, of course, everybody knows that skeletal muscle makes you wiggle, but that's not all it does. It also holds you still, right? So if that doesn't make sense to you, could I be standing in front of you, right, if I were quadriplegic? No, of course. I have to have muscular contraction in my abs and my back and my thighs and my butt and my neck in order to hold myself up. So postural stability is skeletal muscle contraction. What it is it's just a different type of skeletal muscle contraction. And so if you guys did this in a lab this week, you will know that postural stability will be isometric, which you're not trying to change the length, right? And wiggling is isotonic, and we'll learn those names and we'll figure out what that means in just a second, or actually next time. And then sometimes skeletal muscle is less about posture and less about wiggling and more about support. And so what do I mean by support? I mean that sometimes you're just holding things in. For instance, there's skeletal muscle right here that keeps your junk from falling out, right? There's a big floor of skeletal muscle right there called the pubococcygeal muscles, right? Or the pubococcygeal muscles, and the pubococcygeal muscles, my golden attacked me before I left this morning, and I'm so hairy. Pubococcygeal muscles, those muscles, you gotta have stuff going in and out, so there has to be a hole there, right? But you need for your junk to not fall out, so there's a layer of muscles down there. Keeping those intact is a really good idea, because what can happen if you have a whole bunch of big-headed babies in a row? Yeah, you can rip all those muscles and then your uterus is going to hang down here. Yeah? And your urinary bladder. It's called uterine prolapse or urinary bladder prolapse. That is what I think we should be teaching our adolescents to keep them from having sex too young, right? You have sex, your junk will fall out, honestly, right? So I'd be like, oh, no, let's just shake hands. Okay, so I have an anatomy class that's half full of the super smarty teenagers who are taking, and don't think I didn't tell them that. And I show them grody pictures all the time. I'm like, oh my God, and pregnancy, oh, it destroys everything. Your boobs are never going to be in the same place again. And the girls are like, oh. Okay. The dudes are like, I don't see how this affects me, really. I'm not, no. Okay, guarding openings. In some places, skeletal muscle sphincters will guard openings like at the distal end of your digestive tract and the distal end of your urinary tract. You are in voluntary control of those, which is why you're not peeing down your leg right now or pooping down your leg. Heat production. When you contract a skeletal muscle, what you are actually doing, and we'll figure this out in detail, is you're taking chemical energy in the form of ATP and converting it to kinetic energy in the form of muscle contraction. Yes? And we know that energy conversions tend to be very inefficient. And because they are inefficient, you lose what as a byproduct? You lose heat as a byproduct, which is why you can use shivering to heat up your body, which is why if you go and run outside when it's 45 degrees, your body doesn't go to 45, but it doesn't even stay at 98.6, right? It goes up to like 102, right? Because it's like this energy conversion gives off heat as a byproduct. In addition, skeletal muscle, if you want a joint to be stable, you need to strengthen the muscles around the joint. So if you have a bad back, your doctor doesn't say, hey, don't ever use your back again. They say you need to strengthen the muscles in your back and in the antagonistic muscles in your abs. And then skeletal muscle also keeps your juices moving inside. Now, your heart is, of course, primarily responsible for your blood moving through your arteries, but moving through your veins, it's primarily skeletal muscle contraction that actually helps to return it. And then the lymphatic system doesn't have a heart at all. And since it doesn't have a heart at all, you're going to have to squish it with skeletal muscle to keep lymph moving. And the lymphatic system is primarily about draining excess tissue fluid, right? And also about immune system. So you'll actually be less bloated, right? And also have a better immune system if you are more active for multiple reasons. That's one of them. Okay, so let's just get briefly started on skeletal muscle and have you guys understand a really important part about skeletal muscle. First off, let me show you something. So here's a skeletal muscle histology slide that also shows you the innervation of skeletal muscle fibers. And here's something that's really interesting that I want you to understand that's different, and I want you to conceptualize it. So with skeletal muscle fibers, and there's 1, 2, 3, 4, 5, and a little bit of 6, right? There. With skeletal muscle fibers, in my biceps brachii, I didn't make this number up, I actually looked it up yesterday, so I'm not even lying this time. I have usually around 250,000 muscle fibers in my biceps brachii. And in order for those 250,000 fibers to contract every single one needs innervation from the nervous system. So this is a very, very highly innervated tissue. If I ever lose innervation of a single fiber, that fiber is paralyzed unless and until I actually re-innervate it. That means that right here, what you're actually seeing is this is the axon telodendria from a neuron, right? And this one is actually forming a synapse right here onto this muscle. It's called a neuromuscular junction, and it's a special type of synapse between a neuron and a muscle cell, and we're going to look at it in detail. Here's another one forming another neuromuscular junction. What I'm telling you is if this one ever gets broken or cut or torn, this muscle fiber is paralyzed, right? That means that, for instance, if I ever had nerve damage, can you understand how nerve damage can manifest in paralysis or muscle weakness? Yeah? Because if it's muscle weakness, what does that mean? You did what to some fibers and not all? You lost innervation to some and you didn't lose innervation to all. Does that make sense? So that's really important about skeletal muscle, and it's totally different than cardiac muscle. How does cardiac muscle get around the need for individual innervation? Gap junctions, exactly. So if you guys were cardiac muscle cells and I gave her a command from the nervous system, she could tell you and you and you and you and you. So this is an incredibly highly innervated tissue, and you have to innervate every single muscle fiber. And I don't know, I want to know like how many muscle fibers, I didn't look this up, how many muscle fibers are in like a huge muscle like the gluteus maximus or something like that. But there's 250,000 apparently in this one. It can go up to 400,000 in people who inherently have bigger muscles. You really don't get any new muscle fibers after adulthood or after embryonic development, but some people just inherently have more, like Serena Williams, right? You've like, look at her family, right? Those are some muscular people, like inherently muscular. She also works her butt off, right? But in addition, right, when they were born, they were not like the twiggiest little kids in the class, right? They weren't. Okay. So let's talk about terminology a little bit. Each muscle fiber actually comes into contact with the nervous system at a place called the neuromuscular junction. So this right here is the axon of a motor neuron. Before we go any further, let's make sure that terminology is okay with you. What do I mean by motor neuron? You learned this in anatomy. Yeah, I'm going to ask you that next, so hold on to that one. What is motor neuron? You know what a neuron is. I know you do. It is going to control muscles or glands. It's going to control an effector. So that's a motor neuron. Now, what if I told you it was a somatic motor neuron? What is that telling you? Do you remember? Somatic specifically is voluntary control of one specific tissue. skeletal muscle, voluntary control of skeletal muscle. You learn this in anatomy. You just got to brush it off, right? So this is a somatic motor neuron. The site at which it communicates with a skeletal muscle fiber is called the neuromuscular junction. It's a type of synapse, only the postsynaptic cell is a muscle cell instead of a neuron, but the presynaptic cell will work exactly the same as we expected it to. Action potential, exocytosis, neurotransmitter, bind to receptor, the whole thing. It'll be exactly the same. Is that okay? Okay, so this is a neuromuscular junction. And then in addition to being very, very highly innervated, skeletal muscle is also very, very vascular. So this figure is a type of staining in which they stained not for the skeletal muscle fiber, which is in here and looks kind of ghosted, but they stained for the blood vessels that are around it. So what I want you to understand is that every single one of those 250,000 muscle fibers in my biceps brachii has a sweater of capillaries around it, right? So it's an incredibly vascular tissue, highly innervated, incredibly vascular. Now I know that you know, but I want you to verbalize it. Why? Why is this an incredibly vascular tissue? Because, yeah, it takes boatloads of energy, so it needs lots and lots of oxygen and lots of nutrients. We will see how incredibly energetically expensive it is. It's more expensive than muscle. Go ahead.
Why, when like you get electrocuted, can you see each of the? I'm assuming it's veins.
Nerves.
Nerves. So we had a patient that like grabbed something and it electrocuted them, and like you could see in their foot where it was because it went into their hands and out their feet, and you could see each one. So that you're seeing the nerves through it.
Yeah. So what happens? I think the last time I looked this up, it's a little rusty, but what I think you're seeing is you get a massive surge of electricity that goes down the individual nerve fibers, and when it goes down the individual nerve fibers at high heat and high speed, it actually ends up causing scar tissue that shows on the peripheral, just only the superficial nerve fibers that show on the skin. It also happened internally, but you can't see that occurring. So yeah, that occurs on the skin when somebody gets electrocuted, sometimes if it stays superficial. So it's kind of cool looking though.
Yeah.
Are they okay?
They're okay.
They were better before.
I was just confused as to like what you were exactly seeing.
It's actually nerve fibers that you're seeing. The nerve fibers are sort of like burning into the skin, and it'll go away eventually, but
It's weird how it burns the muscle too.
yeah, it's because these are electrically active tissues, and what you end up doing is you end up delivering such a high voltage that it causes massive and also really hot electrical stimuli super frequently, and it ends up like damaging the muscle and the nerve. Sometimes they can regenerate though, not always. Okay, each muscle fiber is surrounded by blood capillaries. You need lots of oxygen and nutrients for contraction. Okay, so next time what we are going to do after you have your quiz over synapses is we're going to build all the way down to a sarcomere, but we're going to do the building quickly so that we can get it wiggling. Okay, which means I need you guys to make yourselves comfortable with it by reviewing sarcomere structure before we get here. So we can go through structure fast to get to function. Okay, I will see you guys on Tuesday. I have office hours after this for those of you who want to come look over your